Good afternoon. Having done meiosis, we now need to look at how inheritance actually happens. And the purpose of this particular lesson is to look at something firstly called monohybrid inheritance, and secondly, dihybrid inheritance. Monohybrid inheritance is where you are inheriting one particular characteristic, such as eye colour or height. Dihybrid inheritance is where you're inheriting two characteristics from your parents, and we're looking at how the different combinations of, for example, different coloured eyes might match up with different heights. The first, monohybrid cross, is the simpler, but dihybrid cross isn't particularly complicated and follows on nicely from it. Before we begin, we need to look at a few definitions relating to the subject of inheritance. This presentation, incidentally, is on the Peterhouse Revision website, so you can always follow it up if you wanted to look at it more carefully without the background light reflections coming in from the window that we've got today. You can do so. You look in the biology folder, the A-level biology folder, and there's a subfolder called PowerPoint presentations, and you'll find this in that folder. So, definitions. We've already looked at some of these. The gene is obviously part of a chromosome that codes for a particular protein. The locus, you drew a diagram showing a picture of a chromosome with the locus, where it is the position of any particular gene on that chromosome. An allele is one of several different forms of the gene concerned. So in this particular case, we're talking eye colour. So the gene is the eye colour gene, but the alleles are either blue or brown. You can only express one allele at a time, except in certain circumstances. The majority of inheritance is where an allele is either exposed in one way or expressed in another. So, for example, here we're saying eye colour doesn't constantly change even if you inherit two different alleles from your parents. So you may inherit the blue allele from your father and the brown allele from your mother, but your eyes are either blue or brown. You don't get shades in between. There are exceptions to that rule, which we'll talk about a bit later on. So normally, one of the alleles is dominant over the other, and that is the one that will be expressed in the phenotype. The phenotype is what the genotype looks like in real life. The genotype is the combination of different alleles. So one allele is dominant, therefore the other one is recessive. It doesn't get expressed in the physical appearance, but it may be passed on to the next generation. So a good example is the allele for eye colour, where we're talking about blue eyes. The blue allele is the recessive allele, so you may have brown eyes, but you may well be carrying the blue allele from your father or your mother, which you can then pass on to the next generation. As I said just now, there are a few exceptions to that. Occasionally, you get a situation of what is called codominance. Both alleles show in the physical appearance. So this is a simple example. You've got two different alleles for flower colour. One is red and one is white. And if the flower has both those alleles, the flower is then pink. If it only has red alleles, it's red. And if it only has white alleles, it's white. The term homozygous means that the alleles are the same on both chromosomes. So going back to the eye colour again, remember you're taking a chromosome from your mother and a chromosome from your father. And if the allele for eye colour is the allele for brown on both your mother's and your father's chromosome, then you are homozygous for that characteristic. If, on the other hand, the alleles are different, say for example you inherit the brown allele from your mother and the blue allele from your father, then you are heterozygous for eye colour. And I mentioned just now the two different words. The first was phenotype. And the phenotype is how the alleles express themselves in the physical characteristics of the person or the living thing. So the phenotype in the case of eye colours is the colour, so brown eyes or blue eyes is the phenotype. Whereas the genotype is the combination of alleles that make it up. So it might be homozygous for brown eyes, in which case it's got both the brown alleles, one from each parent. It might be homozygous for blue eyes, in which case it's got the recessive blue allele from each parent. Or it might be heterozygous, in which case it's got one allele from each parent, the brown one from one parent and the blue one from the other. So now moving on to monohybrid crosses. Monohybrid, remember as I said at the beginning, is the inheritance of one characteristic. Only one gene, for example in this case eye colour. Now, one important rule to follow is that whenever you do a genetics question, an inheritance question, even if it doesn't say it in the question, as a rule of thumb, you should actually outline the cross, the genetic cross, in a particular way. And I'm going to go through that now. So when you come to do questions on this, in general, follow the rules, even if it's not specifically implied in the question. Now, in genetic crosses, we talk about alleles, not in a cumbersome way of writing words like brown for eye colour or blue for eye colour. We allocate the different alleles a letter, and as a general rule, we use capital letters for the dominant characteristic. So let B represent brown eyes, and the parent is homozygous for brown eyes. So that parent has inherited two alleles, one from each of their parents, and each of those alleles is capital B. Little b is the recessive allele. You always, for the recessive allele, have the small letter, the lowercase letter of whatever you used for the dominant one. So big B always has a small b. In this case, it's a little confusing because, of course, the recessive colour for eyes is blue, and so therefore what we've got here is two B's, capital B for brown and small b for blue. But remember, the important thing is you give the letter according to the dominant characteristic, and then the lowercase letter that goes with that is automatically recognised as the recessive characteristic. So what are the parent phenotypes? If the parent is homozygous, it means it's got the same allele on each of its chromosomes. The parent genotypes, exactly the same thing. So, let's have a look. So, the parent phenotypes, we're talking about homozygous parents here. So, the parent phenotypes, this one is going to be brown, and this parent is also going to be homozygous, but it's blue instead. So those are the phenotypes of the parents in this cross. The parental genotypes, we're told here that the parent is homozygous. So therefore, the parent will have that allele on one of its chromosomes and that allele on the other of its chromosomes. Homozygous, both the same, one of these alleles on each of the chromosomes in the homologous pair. And the blue-eyed parent is going to be exactly the same like that. Now, when gametes are formed, you'll know from meiosis that we did just recently, what happens is that the number of chromosomes halves to make the gametes. So instead of having a homologous pair of chromosomes, you've got one chromosome only in the gametes. So in this case, the gametes are going to be made by splitting this into two separate bees like that. So the gametes for this particular genetic cross are, for one parent, are going to be all capital B's. Each of these represents a gamete. So that is a gamete, that's another gamete. So all the gametes coming from this particular parent are going to be capital B's. All the gametes from this parent are going to be small b's. Now, one thing that isn't often told in the textbook, but I find it very helpful, even though we're not at this stage talking about the sex of the individuals concerned, later on in the topic when we talk about things which are related to the sex chromosomes, in other words, sex-linked characteristics, it's sometimes important to know which is the male and which is the female. So I normally take the top of this box and I put a sign for male there and a sign for female here. It's not relevant at all in this particular example, but it's worth doing as a matter of course. So now what we say is we've got these sets of gametes. The father has provided gametes, all which have got the capital B, the dominant allele, and the mother has provided alleles that have got the recessive gene, the recessive allele, small b. Now, fertilization happens. So we've got sperm fertilizing eggs. And every one is going to combine again to make the zygote. So you get that. So that allele and that allele goes to there, that allele and that allele goes to there, and so on. Now the genotype is that. Now, the colour of this person's eyes is going to be, remember which is the dominant and which is the recessive allele. So what colour eyes is this person going to have for the phenotype? And the answer is brown. It doesn't matter that they're all carrying the blue allele as well. That's always going to be smothered because it's recessive. The genotype is going to be something called I can't spell. Heterozygous. So it's heterozygous for brown eyes. And the ratio, in this case, is irrelevant because there's only one type. Ratios come where you've got two different representations. Some have got blue eyes, some have got brown eyes. How many have got blue, how many have got brown? That gives you a ratio. So in this particular example, that is not relevant. So, quick recap. Homozygous parents. The dominant allele there, the recessive allele there. This person had brown eyes, this person had blue eyes. This one is the male, so it's producing sperm, and each of the sperm have one of the blue alleles in it. This is the mother going to produce the eggs, and each of the eggs is going to have one allele in it. Egg and sperm combine during fertilization, so you get that combination at the end, and they're all the same. Before we move on to the dihybrid cross, I want now to change this slightly. And I want to talk not about the F1 generation, which incidentally is correctly named the first filial generation. What I'm going to look at is the F2 generation, is where you get two of these individuals, who were the F1 F1 generation being crossed together and reproducing. And that is the second filial generation, or F2. In this case, both parents are no longer homozygous, they are heterozygous. So both parents are heterozygous, both the same. So the parent phenotypes, going back through the same routine, the parent phenotypes are both brown. Remember, brown is the dominant colour, so if they've got brown and blue alleles, they're going to be brown. The parent genotypes, all that. Remember, that was the previous answer in here. They're all heterozygous, showing both of the genes. So the gametes, it works exactly the same way as before. Meiosis is going to split those alleles, and so the possible gametes in this cross are B and little b. So once again, we build up this diagram, which incidentally I haven't mentioned is called a Punnett square. I assume after Mr. Punnett, who perhaps invented it. So once again, let's put our details in here. Again, not necessary for this particular cross, but quite useful to get into the habit of doing it. So, there's the alleles, two possible alleles now, from one parent, either that one or that one. There's the parent, there's the two alleles. And here's the other parent, and again, the two alleles. So now, fit them together. Think about it. What's going in there? What's going in there? What's going in there? What's going in there? So now we've got some differences. This one, and these two are all different. Let's have a look at the genotypes. So what are the possible genotypes? These are they. So the possible genotypes is this one, which is that. This one, which is the same here. And finally this one. So there are three different genotypes. How many phenotypes are there? The way that this is expressed in the physical appearance. Obviously, this one is going to have brown eyes because it hasn't got the blue allele at all. So that's going to be a brown. What colour is that one going to be? Again, because it's got the capital, that's the dominant one, so that's going to be in the phenotype. So this is going to be brown as well. So you don't need to rewrite it. And the phenotype of this one, two recessive alleles, is going to be blue. Now the ratio comes in. The ratio of the dominant characteristic to the recessive one. 1, 2, 3. Brown, brown, brown. And 1 blue eyes. That's an extremely important ratio that you'll come across a lot in monohybrid inheritance. Whenever you get a ratio of two different phenotypes of 3 to 1, you know you're looking at a monohybrid inheritance, a monohybrid cross. Okay, so that's the basic simple outline of a monohybrid cross, going through from the parents, which were pure breeding, homozygous, to create an F1 generation when you make them together, and then the F2 generation where you take two offspring from the F1 generation and they are mated together. And you always, in a monohybrid cross for the F2 generation, if you're breeding, if you're crossing two of the original F1 generation offspring, you're always going to get 3 to 1 as your ratio. Now, moving on to the dihybrid cross. Briefly, this is not in the syllabus, but very briefly, there was a man called Gregor Mendel. He was a monk, incidentally, and he looked at pea seeds and pea plants back a couple of hundred years ago in his monastery. And what he was doing is he was looking at the colour of the pea seeds, and he found there were several different colours. He also looked at the shape of the pea seeds and found they were either round or wrinkly. He also looked at the colour of the flowers, and he also looked at the size of the pea plant. We talked about variation at the last lesson of last term, and when we were talking about the effect of plant hormones on the height of plants, and I said that the peas, incidentally, were of two sorts. They're either tall or short. Dihybrid is showing two characteristics in one genetic cross. So Mendel was looking at peas. He was looking not just at the colour of the seeds, he was looking at how tall they were in the same experiment. He didn't have any idea about genetics. He was just doing simple experiments in his plot of land, and he came to conclusions not understanding anything about genetics. But he was the father of what is now referred to as Mendelian genetics. So, here we've got a couple of examples of peas. Round seeds, facts first of all, round seeds are dominant over wrinkled seeds. Yellow seeds are dominant over green seeds. So what are we going to call the dominant allele for shape? Remember, here's the shape, and the dominant characteristic is a letter which best represents the shape of the phenotype, or the phenotype characteristic. So it will be sensible for the dominant allele for shape to be R for round. Wrinkled, which you would think might be a W, remember from the earlier section I said that the letter is always the same as this, but it's just a lowercase letter. So for wrinkled is small r. Dominant allele for colour, and the recessive allele for colour, not G for green, but small y because it's recessive to the yellow. Now it gets a bit more complicated with a dihybrid cross. Don't forget we're looking at two different characteristics. So there's two pairs of chromosomes with two alleles. So the parent phenotypes, if we are talking here about homozygous individuals. So if the P is homozygous for the shape, let's do this first of all. What colour and what shape are the dominant parents going to be? They're all going to be round, because round pods are dominant over wrinkled pods, and they're all going to be yellow rather, because because yellow is the dominant colour, the dominant allele over green. So the parent phenotypes, they're all round and yellow. The parent genotypes, remember we've got two different chromosomes now, one showing the shape and one showing the colour. So we're not now going to have just two alleles, we're going to have four. Two representing the shape and two representing the colour. So the parent genotype of pure breeding yellow peas that are round are going to be this. Because pure breeding round, each chromosome has a capital letter, a dominant allele. Pure breeding for yellow, it's going to be like that. This is being crossed with the recessive homozygous, just like we did with the monohybrid cross, where we crossed a brown eye on a blue-eyed person. So the shape of the recessive phenotype is going to be wrinkled, and it's going to be green. Because wrinkled is recessive to round, and green is recessive to yellow. So the phenotype of the pure breeding recessive parent is going to be if this had any of these capital letters in, it wouldn't be wrinkled and green because it can't have any dominant alleles to show the recessive phenotypes. The gametes, we've got more space for gametes here. Once again, we start in the usual way. Each gamete is going to have two alleles, one from each of the chromosomes. So here it's going to have a shape allele and a colour allele. So each allele is going to be like that. Here again, each allele is going to have a shape and a colour. Like that. Now I'm not going to spend lots of time filling in the whole thing. It will get very boring because you can see that they're all going to be the same. They're all going to be... When you write it down, don't mix the letters up, keep them together. So don't put big R, big Y, little y. Keep the letters together. So they're all going to be like that. Whichever one you look at, that one, R, big Y, little y. They're all going to be the same. So, don't worry about this. This is for the next example. So these are the alleles, these are the genotypes of the F1 generation. And what colour and shape are they all going to be? For the shape, they're all going to be round. For the colour, they're all going to be yellow. But they're all heterozygous for both of the characteristics. So they are heterozygous, but they are round and yellow. Now, the only thing you need to carry forward to the next diagram, showing the F2 generation, is this. So we'll put that over here. And then rub everything else out. And start again. So, here are the parents. We're crossing two of these together. So mating that. Male plant, female plant. So, to produce the gametes, this time, there are 4 different possibilities. Remember that each gamete has got to contain one allele for the shape and one allele for the colour. So each gamete has got to contain either a capital R or a small r, and a capital Y or a small y. So if you think about that, it's very straightforward. So one of the alleles, capital R, capital Y. Another one of the alleles, capital R, little y. Another one of the alleles, little r, big Y. And the final allele, little r, little y. And that's going to go down this side as well, because remember the parents were identical. Now, filling this in is more complicated. You have to think a bit. Because here you've got quite a few different variations. And so on. I'll leave you to fill the rest of this in yourself. You can draw yourself a Punnett square and fill it in. So I suggest what you do is just pause the video for a minute and do it. See if you can work it all out. So pause the video. When you've done it, switch it back on again, and I will now carry on putting the rest in. So now hopefully you've done this for yourself. Nearly there. Okay, now, we now come to the final ratio of this particular cross. We've now not got just two possibilities like we have with the monohybrid, we've got four possible outcomes of phenotypes. First of all, we're going to look at the number of individuals that have got, that are round and yellow. So remember, to be round and yellow, you've got to have a capital of each letter. So you've got to have at least one capital R and at least one capital Y. It doesn't matter if you've got two of them, but you must have at least one of each. So let's cross them out. There's one. There's one. It's got one of each. Two of one, one of the other. This one. This one. This one. Not this one, because there's no capital Y. This one. Not this one, because there's no capital Y. This one, not this one, because there's no capital R. Not this one, because there's no capital R. This one, not this one, not this one, not this one. So how many have we got? 1, 2, 3, 4, 5, 6, 7, 8, 9. The next possibility is round. I hope you can see this. Round and green means it's got to have a capital R, but it mustn't have a capital Y. So there we go. One there. One. There. Two. There. Three. That's the only three. They're the only ones that have got a capital R but no capital Ys. That one hasn't, that one hasn't. So round and green is 3. wrinkled and yellow. This one has got no capital R's in it, and it has to have at least one capital Y. So we'll cross them in a different way. Let's cross them this way. This one, capital Y. This one, capital Y. This one, capital Y. So again, it's 3. And in a way that you can see this at the bottom, wrinkled and green. We've only got one left. Look, there we are. To 1. So, we've now got a 9 to 3 to 3 to 1 ratio, which is the standard ratio for when you're talking about a dihybrid F2 generation cross. 2 different characteristics, in this case, the shape of the seed and the colour of the seed. Each parent is heterozygous. Each gamete contains 1 allele for shape and 1 allele for colour. So there, and there. Shape, 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 colour, 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 colour. Blend them all together. In order to show in the phenotype the dominant characteristic, it's got to have at least 1 dominant allele for the shape and 1 dominant allele for the colour. So it's got to have a capital R and a small r, a big capital Y. For round, it mustn't have a capital for the shape. For wrinkled, it mustn't, for yellow rather, it mustn't have, for wrinkled rather, it mustn't have a character, the dominant allele for the shape. It's got the dominant allele for the colour, so it's got a capital Y. And finally, the one that's going to be recessive is homozygous recessive, because it's got both the same allele for recessive characteristic for the shape and for the colour. So if you were to look at the genotypes, you've only got one that is homozygous for the colour and the shape. That's that one, because it hasn't got a mixture of the upper and lower case. You've only got one down here that is homozygous for both the colour and the shape. Everything else is heterozygous for something. So just recapping. Okay. That basically concludes what I want to say. The assignment that goes with this, which you'll find on the Google Classroom, is a number of different problems relating to both monohybrid and dihybrid cross. Please attempt those questions, do the notes that I've asked you to do, get the assignments in by the deadline, but please do contact me in the meantime if you have an issue.